Well, good evening everybody. My name's Bob Hill. I'm the Executive Dean of the Faculty of Sciences here at the University of Adelaide. I'd like to commence by acknowledging the Kaurna people, the original custodians of the Adelaide Plains and the land on which the university's campuses at North Terrace, Waite, Roseworthy and Thebarton are built. So welcome everybody to tonight's Research Tuesday event. As many of you already know, this year's the Research Tuesdays program is incorporating a series of inaugural lectures. These lectures are held to mark a major milestone in our researchers' careers, such as achieving the rank of professor or being appointed to another senior position. Tonight we welcome Professor Bronwyn Gillanders, who is the Deputy Director of the university's Environment Institute. So, a bit of background about Bronwyn. She received her Bachelor of Science from the University of Canterbury in New Zealand and a Master's degree from the University of Otago before moving to the University of Sydney, where she undertook her PhD in Biological Sciences. Following her PhD, she spent a year working in government in New South Wales fisheries before taking up an ARC fellowship at the University of Sydney. She moved to the University of Adelaide in 2001 to take up an ARC Queen Elizabeth II Fellowship and commenced an academic tenurable position in 2006. She held an ARC Future Fellowship. She's mentored around 10 postdoctoral fellows, 20 PhD students and about 30 honours students, and has over 100 research publications. Most recently, Bronwyn's been the driving force in the Spencer Gulf Ecosystem and Development Initiative, which is a very important and very large-scale initiative for the university, based obviously around Spencer Gulf. It's my great pleasure tonight to welcome Bronwyn to the stage.
Quantifying the impact of human and environmental disturbance on populations is difficult because we generally lack many of the historic ecological baselines. Whoops, sorry. If we have a look at trying to tease apart some of the ecological responses of a changing environment, we find that difficult. Here we can see that there's really a paucity of long-term ecological data sets in aquatic systems. We've got a lot of information from ice cores and the like. We have a reasonable amount of long-term data sets from terrestrial environments, and I'll show you an example shortly. Down here we have very few from marine environments. Most of you will be aware of the long-term ecological data that can be obtained from tree growth rings, and you can see that here in the bottom picture here, where we've managed to reconstruct precipitation records over just over 200 years of data, and it's shown here in the black line with the red line representing the modern day precipitation. This is a tree ring record that we've obtained from the east coast of North America. But 70% of our planet is actually water, so it should really be called planet ocean rather than planet Earth. And for many of those systems, we really do lack long-term data sets. An example that's often used is from coral reefs, where we can take coral cores through the reef and get quite extensive information. And this paper here is from over 10 years ago from colleagues in ANU that uses barium calcium ratios throughout the coral from the inner Great Barrier Reef to establish a record of sediment flux at weekly resolution. This is their data here, where the green line represents the barium calcium ratio, and again it's over about 250 years. And what I really want you to note is from about the time of European settlement, we get much greater fluxes of sediment into that reef system. The black line here represents the Burdekin River discharge, and if we just focus in on a 20-year period, shown here, we can see that several coral cores shown in the different red and green colours here actually are quite similar to one another, and that they correlate strongly with this Burdekin River flow. In temperate systems, we often don't have coral cores or other long-term records. But we do have a range of calcified structures that exist in those systems in which we can get some, perhaps not 200-year records, but certainly some historical data from. And here in this image here is some of the structures that my group works on. We've got the air bones or the otoliths of fish. Here in the middle we have seal teeth. We've got shark vertebrae. And we've got up the top here bivalve shells. So many of these calcified structures are similar to tree growth rings. They encapsulate the entire life of the organism, and you can see the birth part of the organism is right in the centre, and then the most recent life history component is at the edge of the structure when it was killed. They also, as you can see, have got the growth increments. In this case, they're annual. The width of these growth increments can change depending on the environmental conditions, and so they're sensitive to different environments. And many of these other calcified structures or organisms that have calcified structures are much more broadly distributed than, say, corals are. And the other thing that's important here is we can get collections back through time. So I've really just put this in to remind myself to say that while some of these structures are in direct contact with seawater, some of the others are not. So here we can see the otoliths or the air bones of the fish, which are encapsulated within the inner air cavity. You can see what they look like. They're species specific, so the shape of them actually varies depending on the species that they come from, which is also important for some of the work that I'll show you. So we can get modern samples where we know the time of capture. In many institutions, there's also historic archived samples, and these actually come with some immaculately written data on the information related to the fish, but often in these little brown envelopes with this one marked on His Majesty's service, there's a pair of otoliths that have been collected, in this case back in the 1950s, 1952. Now these samples are often tucked away in organisations, or in some cases have actually been lost because they've been thrown out, thinking that nobody would want to use them. The other place we can get some of our samples from are from Indigenous middens, and here you can see a range of samples taken from Indigenous middens. I've put what the modern ones look like. These are from Mulloway, and here you can see well-preserved samples moving through to less well-preserved, or in this, the ones on the right are actually being burnt. So to get the growth information out of these or other information, we usually would take a transverse section, and you can see this here from modern Mulloway otoliths as well as the midden ones. In the case of the burnt ones, we actually get no information or no useful information. And again, here you can see the beautiful growth increments in the Mulloway otolith from a midden. We can measure not only the age or estimate the age of the fish, we can measure the width of the growth increments to get information on the environment, and we can also measure the trace elements and in some cases isotopes within these structures to get information on the environment. So here, the premise behind using these structures as environmental recorders is obviously that they reflect the water, the salinity, the temperature, the trace elements in the water at the time when the material was laid down. But in the case of fish otoliths, there's actually some discrimination along the way, so we don't always get one-to-one relationships. So in my laboratory, we've undertaken a whole range of experiments in the laboratory setting where we can control things like temperature and salinity and look at how that might affect growth and trace elements within the air bones. We've also done work that suggested that the majority of the trace elements that we see in the otoliths have come from the water and not from their food. And we've also used the field and as a natural laboratory. In this case here, we're out at White Island in northern New Zealand, which is an active volcano, and out in the water you can see these bubbles seeping up. So we've got carbon dioxide gradients which reflect the pH in the environment. So we can use these as a natural laboratory to have a look at how the trace elements in the air bones may change as we move across that gradient. And we've also done work where we've got nice salinity gradients like through the Coorong. So for the remainder of the talk, I'm just going to focus on 4 different questions that relate to addressing either environmental or management related issues associated with using calcified structures. The first is going to be using the effect of the atom bomb for tracing water masses. I'll then use an example linking teeth to, or growth in teeth to climate. And then I'm going to focus on several examples where we've used historic data and archaeological otoliths to understand not only the environment and the ecology of the fish, but also the past human occupation. And then finally, because I mentioned it in the flyer, I'll refer to some work we're doing using vertebrae in sharks to look at population structure. So you're probably wondering how on earth a mushroom cloud coming out of the ocean like this from the Bikini Atoll or Bikini Lagoon in 1946 could actually be used in to answer marine ecological questions. So everybody's aware that atomic testing started after World War II and occurred in both hemispheres. If we look, we can see here the distribution. We've got bomb radiocarbon levels here versus time, and you can see prior to the mid-50s the levels were relatively low, and then we've got increases through time in the atmosphere that then drops off as a lot of that testing finished. But we also find that we've got increased levels in ocean waters as well. So the presence of the bomb carbon in the Earth's biosphere can actually be used as a tracer for looking at questions related to carbon mixing and exchange processes. It's essentially a global age marker that's also been incorporated into many of the calcified structures that we look at. And from this, or in this respect, it's actually been referred to as the silver lining in the thermonuclear bomb testing. So while everybody thinks of the negatives associated with nuclear bomb explosions, it has actually provided marine biologists with a global age marker. So this is the research that one of my PhD students, Gretchen Grammer's, been involved in, where she's essentially using the fish otoliths as a temporal record of carbon-14 flux through ocean waters from the surface to around 1,000 metres. And the work's focused here in eastern Australia and New Zealand. And she's predominantly focused on this one species here, big eye ocean perch, off the coast of eastern Australia, but there's also existing collections of several shallow water species and orange roughy that will help us trace the water masses or help us trace what's happening with the radiocarbon across different depths. So if we know when the fish were collected, we've got an edge sample that reflects the time of collection. We can back calculate through to what year the core is, of the fish represents, and by sampling that core region we get also get a historical record. So these are some of the results where we've got bomb carbon on this one axis versus time, and you can see for orange roughy, an incredibly long-lived fish, that prior to the nuclear bomb explosion we're down at relatively low levels, and then once the bomb carbon has occurred, the explosions, we can pick up an increase in orange roughy radiocarbon. So we've got a surface water curve here, the data from our fish that was collected at 400 to 500 metres, which is a mid-water curve, and then the orange roughy, which is a deep water curve from 800 to 1,000 metres where they were collected. If we look at how the slopes of increase change for those three different groups of fish, we can predict that there's been a 5 to 10 year lag in the way bomb radiocarbon is incorporated from the surface waters to the mid waters, and then a further 10 to 20 year lag going from the surface waters to the deeper waters. If we then have a look at the different water masses in those regions, and if I put the fish on the top where they were collected from, basically we've got fish coming from three different water masses, and we can actually use this radiocarbon to trace water masses. If we have a look at the flux from the atmosphere, so this is just putting it back into the broader context, this is what the atmosphere shows. This is a coral record from the tropics, and you can see our surface water fish actually match that record. So we're using these fish air bones to suggest that we've got another means by which we can trace movement of water masses. So if I move to my second example of using calcified structures, here we're using teeth and linking that to climate. Most people will be aware that there's been significant changes in our global climate. We've got changes in temperature as well as ocean circulation, and this can significantly affect the physiology of the organisms and indirectly affect their food availability. Besides modifying things like abundance and energy budgets, we can also see changes in growth. So our research here is using what's been recently renamed the long-nosed fur seal. It's in fact more commonly known as the New Zealand fur seal, and using their teeth to see if we can determine if the growth within the teeth is actually correlated with climate indices. We know that the dimensions of the teeth are related to the body size of the organism. These guys are relatively long-lived, 24, 25 years. They show strong site fidelity in terms of returning to natal and breeding areas. And the annual, or the increments within the teeth have been validated as occurring on an annual basis. And the samples that we're using here actually come from existing collections. So this is what the samples are on slides. They've been decalcified, stained, and sectioned so that we've got beautiful sections. If we look in closer detail, we can see one of these tooth sections here, and if we focus in, we can see there's growth layers within the cementum part of the tooth. So we can use the width, measure the width of these growth increments, and start having a look to see whether we've got any relationship with environmental parameters. Now, I'm not going to go through equations. I've really just put this in here to remind me to say that we use a range of biological variables to come up with the best starting model for predicting growth in these teeth, and then build on these models by exploring some of the environmental information. This graph here shows you the predicted width, it's an index, so it ranges up the top here from 0.2 to minus 0.2. I've put a line through the centre here, and the easiest way to think about these data, this is what the growth of these teeth are doing through time, and anything that's above that 0 line is a good growth year, whereas anything that's below that 0 line is a bad growth year. So in this case, you can see 1988 here was a great year, and there's a few other years here. And you can also potentially see that 2001 here was a poor year. If we correlate that to the Southern Oscillation Index, so that's a global atmospheric ocean phenomena, and also to sea surface temperature, we can see that we get some quite nice relationships, particularly with sea surface temperature. And in this case, it's negatively correlated with sea surface temperature. If we have a look at the males, and here we've got a much shorter chronology, so the males are actually, or the samples that we had were actually younger than the females. Again, we can have a look and see when we've got good growth years up here and when we've got bad growth years. So just to summarise for the teeth information, sea surface temperature is negatively impacting fur seal growth, and with predicted increases in ocean temperature, we can use these historical climate growth relations to start having a look and making predictions around what impacts might be into the future. If you notice the axis, because these are samples that were already existing, our latest samples come from 2001. If there was further sampling undertaken, we could actually extend the length of that chronology by another 15 or so years. So the possible explanations for what might be influencing these growth increments in the teeth, that it could be a direct influence of temperature on the physiology of the organism, or it might be an indirect effect such that it's influencing the food that the fur seals are feeding on. So the third part I wanted to focus on is what otoliths or air bones of fish can tell us about humans and the environment. Now, I'm an ecologist, so I typically think about the environment and think that we can use these structures to start looking at changes in climate and ecosystems, or we can start to have a look at changes in the biology of the organism or the ecology. But I have a student that has an archaeology background, and she really did open my eyes as to the sorts of information we could potentially also get from some of these structures about the human population. So many of the samples from Indigenous middens are there because they were used as food by Indigenous populations, and so they automatically give us an indication of the diet, and in some cases that can also indicate trade routes and the like if that species wasn't known from the area. The other thing that I was really fascinated about was you can also get information on the seasonality of site usage. So was the site potentially used year-round, or was it just a warm weather site? And I'll show you an example of that shortly. The other information you can get is on fishing methods. So depending on the size, we can use these air bones to reconstruct the size of the fish that that Indigenous people fished, as well as the age of these fish. And depending on whether they're all large fish or whether they're across a range of different sizes, we can get estimates of the likely fishing methods that they used. And also, I showed you earlier the preservation of some of the otoliths. If they're burnt otoliths, that gives us an idea about some of the cooking or processing methods that might have been used. I'm only going to show you one example around humans, but I really did find it fascinating, the diversity of information that fish air bones could tell about humans. So this is the bit that focuses on the seasonality of site usage. So by looking at the edge increment, And here you can see we've got a wide edge increment that's likely indicative of a warm season, and over here we've got the increment right on the edge, so that suggests it was caught at a different time of year. So by knowing a bit about the biology of the species today, using the modern growth information, we can estimate the approximate month of death, remembering that these archaeological samples, and yes, the site was only occupied for a short period of time, really they've come from the last 2,500 years, but over a whole range of years. So these are samples that we've used to look at seasonality of site usage down in the Coorong area, and in this case the majority of the fish were actually harvested during the warmer months, suggesting that they may have focused on the fish, this particular species, as part of their diet during the warmer months. The other thing we can get an indication from using the fish air bones is of the age structure of the population. So this is data that was taken from the Murray-Darling Basin. It's from golden perch, or callop, some people may know it as. And these are modern day data, and in this example you can see that very few fish lived over 20 years. And we might think of this as the norm if we knew no different. But in fact, if we look at some of the fossil otoliths down the bottom here, and the age of those otoliths, exactly the same species, we can see that a significant number of fish were much older than 20 years. And so this is what we'd refer to as a shifted baseline. If we know no different, we'll think that that's what naturally occurs. So what you can see here is significant age truncation up in this top graph, where we really get fish over 20 years. And what's been attributed to the demise of many of these inland fish is commercial fishing of the early European settlers. Now, Heidi Allaway is a PhD student of ours who's been having a look at some of the historical ecology around the inland fisheries, and I just thought I'd put in a couple of the quotes that she's got, because I think they also raise issues around changing environmental conditions of the past as well. This is a quote from around Murray cod. It comes from 1903, and it suggests that 9 years ago men could make a good living with cross lines and springers. These are types of fishing methods that European settlers brought from Europe and were widely used there. And it was no unusual thing for a man to catch half a tonne a week. And then moving further on, it's with a cycle of normal river levels and judicious fishing therein, is there any reason why those good times should not come again? And then as early as 1903, they're talking about the need for fishing restrictions, and they actually say it's better to inflict this necessary hardship and save the industry then allow the present decimation to go on till all those now engaged in the trade, around 500, find their occupation gone. So that was written in 1903, and we know that that commercial fishery in the river closed in the mid-2003. There was also a comment around golden perch as well as Murray cod, and that relates to the Adelaide market regularly receiving considerable quantities of River Murray fish, and when flooding of the river occurred, these supplies are often so large as to overtax the market. That comes from the 1940s. And so this is a bit of an aside about the historical fishing component. And if we have a look at what data we can get that does go back through time around historical fishing, and these data come from 1900, and it's focused largely on what catch from inland waters was sold at the Adelaide market, and then more recently through statewide production. And the point I really want to show here relates to the percent contribution of inland fisheries to overall South Australian state fisheries. And you can see back in between 1900 and the 1950s, inland fisheries comprised around 40% of the state fishery catches, whereas today they comprise nothing. And this has been used as an example of where the European settlers that came to Australia brought with them some of the fishing methods and their love of fishing, I guess, in freshwater environments rather than the marine waters, and then once they had fished out the inland waters, they've since moved to marine waters. If we then have a look at how we can use these structures to also look at changes in the environment, the report from 1903 fits in nicely with the Federation drought. This is the drought years in the Murray-Darling Basin and their popular name, and a series of photos that relate to the millennium drought of drying waters in that system. As the waters dried, and with the barrages that have been built and the like, there's also a change in salinity in the system. So here we're using the fish air bones, and this is of a black brim, and if we focus in on one growth increment, we can allocate what component of growth was attributed to each of summer, autumn, winter and spring, based on some of the knowledge we know about fish air bones, and that we've sampled them monthly across time, figuring some of this information out. What we've got here is using barium calcium in this case. So barium calcium from within the otoliths has been shown to be linked to changes in salinity as well. And we've got a barium calcium index here for our migrant and our resident fish, so two contingents within a black brim population. And here you can see in green is the salinity. So if you think back to our earlier plot where above that zero line we've got good growth and below we've got bad growth, you can immediately see that when we get higher salinity, and we're talking heading up towards 50 parts per thousand, so above normal marine waters, we get poor growth of black brim. So we've then used a separate data set, the relationships that we've got between those barium calcium indices and salinity to reconstruct salinity on a modern sample, and you can see the reconstructed salinity is pretty good compared to the actual recorded salinity. So we've then thought that because we've got a reasonable relationship, we can use some of the archaeological samples to look at how salinity may have changed in the past. So this is an archaeological black brim otolith, a whole one that's then been sectioned, and we've analysed the barium calcium across the air bone. And here you can see the 19-year record for this particular fish of what's happening. It's got some good years, some bad years. And we can then use that to reconstruct the salinity at the time that the fish was collected. Remembering we don't know the exact date it was collected, it's likely around 500 years old, that particular otolith. And we can start comparing it to the modern day salinity. And what we can see here is that we've got similar minimum values of salinity in that system. So it gets down as low as it did in the past, but we've got much reduced maximum values, so we don't barely go above 30 parts per thousand. So this really was an estuarine system in the past, as opposed to becoming hypersaline like we see today. And remembering this is a 19-year-old fish, so this represents 19 years of salinity data. If we start looking at the overall average and the variance, it's much reduced in the historic sample than what we can see, or in the archaeological sample than what we can see today. So using these archaeological samples, as well as some of the historic samples and the modern day samples, our overall aim is to eventually be able to match up over much longer time periods than what I've been able to show you here, and start having a look at how the environment really has changed through time. So the final example I wanted to just touch on, because it uses another calcified structure, is looking at a pelagic elasmobranch, in this case it's the bronze whaler, and looking at population structure. And it's really important for spatial management of commercially fished species like this particular one to be able to know the size of different stocks and whether they're all the same across all of South Australia, whether it should be managed as multiple stocks or one stock. It's obviously logistically challenging to work on some of these large species, and that's where we can use the vertebrae chemistry of samples that come from the markets. So this is a sectioned vertebrae you can see here, and we've got samples. In this case we're focusing on the Gulf regions of South Australia, and we analyse a transect across the vertebrae, and we can get information on the lifetime of the particular shark based on the signal that it shows over its whole lifetime. But we can also take just the edge data to look at the most recent part of its life history, or we can take an integrated signature that would represent the entire life history of the shark. So these are some of the data that we get from doing that. Because we've got, when it comes to sampling these sharks, it's really difficult to be able to say we want this particular size or this particular age. We start using models that incorporate a whole range of variables, and in this particular case for looking at the mean chemistry, so right across the vertebrae, as well as the edge chemistry. In both cases, most of the variation that we're seeing is either regional, so the region that they were collected from, or it's related to the size of the shark. And there's very little seasonal information from these data. But we don't seem to get consistent patterns around this regional variation, and so that leads us to think that these sharks are not necessarily highly mobile all of the time, but there is some spatial fidelity. When we've looked at the overall profiles across the vertebrae and tried grouping them according to the sex of the shark and also the region it comes from or the size of the shark, we can see that a lot of the movement patterns are highly individualistic. And in this particular case, we've suggested that state-based management appears appropriate, and that's the way this particular species is currently managed. If we can get, for another particular species, we've got a historical data collection, so we can start looking at whether we see changes in some of the population structure across time that might suggest that fishing or other environmental factors are contributing to differences in the way organisms are structured. We're currently augmenting a lot of the data that we're getting from these shark vertebrae with not only demographic and satellite tagging data, but also molecular data using some of the next generation sequencing approaches. So just to summarise, I think I've demonstrated that BOM radiocarbon can be used as a global age marker. In the case of fish, it's often been used to validate ages of long-lived fish, but really as a water tracer or as a tracer of water movements. I've shown you an example of fur seal teeth being used to predict growth and how that might change with a changing climate. And then the archaeological, using archaeological to modern samples, I've demonstrated how you can look at seasonal site usage of Indigenous populations. We've been looking at shifting baselines and changes in fishing and using historic information that many people have not really focused on using that might be available in the archives or through some of the historic newspaper records. We've also seen how you can get a change in environment through time, and in the case of the Coorong, where those fish came from, the archaeological sample compared to the modern day one, and then using vertebrae chemistry to determine population structure. So these calcified structures really are analogous to the flight box recorder that you get in aeroplanes. They record the information and lock it up until we come along and can extract it and what it actually means. None of this work would have been possible without a whole range of colleagues, staff and students, and I'm just acknowledging them here. And if anybody's interested in more information around some of my work, I've just put my website at the bottom here. I'm happy to take any questions.
So Bronwyn, I'm used to organisms like us that we grow, and then at about 14 or 15 years we stop. But these things look like they just keep on growing forever. Is that the plan, or do you have to take into account the actual growth rate through time? Yes,
so they do. They keep on adding thinner layers, but using some of the techniques, we detrend the data so we remove the age-related effects. But they do that. The otoliths, the vertebrae, etc., keep growing through time, even though the organism might slow down.
Okay.
Thanks very much for that, Bronwyn. You showed a very elegant example of how the age structure of the population was truncated by overfishing, and I'm just wondering whether the method is sensitive enough to show the introduction of predators, for example. So could you know, is there a freshwater equivalent of cat and fox introductions?
Yeah, I guess it could show that. The issue here for the couple of species I've looked at in relation to like Murray cod and golden perch is that they're top predators in that system. So, but it'll be interesting to have a look now that they're talking about fur seals going up the river, whether that's impacting some of these populations in terms of predation events. But I guess we'll need to wait a year or two to start having a look, and then you'd also have to start teasing apart the effects of the changing environment as well.
Can you please join me once again in thanking Bronwyn for her talk tonight? And I have a gift here on behalf of the university. Thank you.
Thank you, Brian.
I'd like to let you know about the next Research Tuesday event. On Tuesday the 8th of September we're holding the university finals of the 3-minute thesis competition. We'll have 10 of our PhD students challenged to explain their thesis project in just 3 minutes and using just one PowerPoint slide. It will be a fast-paced event. The 3-minute thesis is a fantastic opportunity to see some of our real budding talent in action. You're certainly welcome to attend. It's on Tuesday the 8th of September and you can register online. So thank you for coming tonight and good night. Thanks.